All right, we're gonna get started in a minute. I'm gonna let people trickle back in. Feel free to read the prompt for the group exercise we're gonna start with. Go for it.
Are other words in different languages easier? For instance, in Spanish, it's not novel. It might be easier to read because every word, like, you can just read it. Whereas in English, if you don't know how it's pronounced or whatever, it's not.
Assume we're saying like in your language, so it would be learning knowledge.
Yeah, but my question is it different in
This is a hypothetical. There is one study I'll show you, but you're asking like the actual science of the question. And I am like,
Oh. Okay, welcome back. We're gonna start this session with a group exercise. So very quickly going back to sort of my key nuisances, as I called them. Keeping these in mind, particularly the first one. Let's talk about a scenario where you want to do research that is going to complicate things. How can you do it? And what factors are you going to control for? How are you going to control for it? So go ahead and discuss with the people near you. If you're online, you're welcome to put it in the chat. Ideas about what we need to control for and how we would maybe design a task to test if reading novel words is more effortful with pupillometry. Just don't do it. That's the perfect answer. That's been my answer so far. Okay, finish your conversations. We're gonna come back together. Okay, so ideas, thoughts. Maybe start with concerns, and then we'll go into ideas of how to deal with these concerns. Sandy, what's your concern? Don't like reading. Reasonable. So my solution in my research has been to avoid research questions like these. But let's pretend that you really care about reading.
Slightly more constructive than Sandy. You want them to be, you don't want to have different length words.
Yeah, yeah. So Raya's brought up that you don't want to have different lengths of words. So we want all our words, our novel words in this experiment to be the same length. So that's one important strategy. That's related to luminance on the screen in particular. Any other thoughts? Sorry? I did. I am recording. Yes, I am recording. Thank you. Any other thoughts about, yep, Tessa?
Regarding the length, the area of because it's luminance as well,
Mhm.
no?
Yeah, so the area of the word on screen, you want to be kept the same. You probably don't want it to be too big. Like I was saying earlier, you want to really have people just looking to one spot on the screen and not looking around as much as you can control for saccades. Another thing, oh,
We are talking about words presented in relation or inside the sentence.
in isolation. Let's stick with isolation for now. Although I have heard from Martha that she's interested in words in a sentence context as well. We can build on this in a moment.
A question, sorry.
Yes.
Like when we talk about balancing out the luminance of the
Mhm.
words, like in this case,
Mhm.
words. Like, it needs to be like a perfect. Like, for example, if some words, like depending on the number of the type of letters that you have in a word, you're gonna have more or less black ink, let's say, on the
Mhm.
screen, right?
Mhm.
Do we need to balance that out as well? Or is that like super fine grading is not that important? Or do we really need to try to get like the same amount of whatever, P and B?
Yes, so the question is, how important is the exact luminance of these words on screen? So if we think about, maybe we could take account of how many pixels are white on a gray screen. What I have done when I do things with visual on screen is I use Adobe Photoshop. They have a tool that I can't think of off the top of my head, unfortunately, but you can check, essentially, you can put in an image and say, tell me the luminance number, and then you can adapt things. There's sort of a simpler solution when I think I'll take one more idea and then we'll look at the actual study that did this, because it sort of gets at your point, and they just sort of found a creative way to work around that type of a problem. Do you want to add another comment?
One with the area of the word fonts which have like typesetting font, which
Yep.
has like same amount of ink or pixel in each letter, so that even if the word size is same, the typesetting because of that font, it's same pixels for each letter. Also for the matching luminance,
Mhm.
there's this shine toolbox which is used in a lot of visual perception guidelines, where you can match luminosity and like all the low-level features of an image. So you don't have to like stick to like words or Photoshop.
Yeah, yeah.
You just put it in and it will match all the. So if you have different images, you can just match the luminance and all these low-level features using this toolbox.
Yeah, that's a really great suggestion. Thank you. So that was a suggestion to use fonts with type settings that will match the number of pixels per letter. And there's a Shine toolbox, which I'm going to ask you to send me the link to the Shine toolbox and we'll share that around. Let's look at, I'm going to switch gears here, bear with me. I'm going to go back to the PowerPoint. Okay. So let's look at the study that actually did this. So I was a peer reviewer for this paper. And when when you get requested for a peer review, for those who don't know, they send you an abstract, like the title and abstract. And then you decide whether you're going to review. And I got this and I was like, there's no way this is going to work, essentially. I read the abstract and I was like, immediately in my mind, I had this sort of mental block against putting words on screen, which is what it sounded like in the abstract. What they did in this study, I think, was very clever. So there are a couple of things they did. First of all, they maintained identical luminance levels across conditions by creating what are called yoked pairs of words. So this study was done in Hebrew. So I just had a moment where I forgot. Anyway, so we can see we have the word snow here on the left, and they have created a non-word for their experimental paradigm by just moving these characters around. So this is a really clever way to essentially, across conditions, the exact same characters are on screen, they're just in different locations, to create words and non-words. The other thing that wasn't brought up in the ideas that I'm really going to stress that I think is really important for their data quality in this paper is before they put the word on screen, they have these X's on screen. This is to say their baseline period has matched luminance as their stimulus period. That is critical. And that is something that we'll see some other examples of too. So for example, if you want to put up images on screen, you can create a scrambled version of that same exact image, have it on screen, and then have it change to the real image. And that is a really, really good strategy to avoiding luminance confounds. And then just to sort of wrap up, their data was quite pretty. When there were real words on screen, there was a larger pupil response, long story short. But we aren't going to spend too much time on this. This is just an example to sort of demonstrate that there are creative ways that you can get around some of the issues we have discussed so far. Yes.
Yeah, but those are non-words.
Yeah. As opposed to novel words. I agree. Yes. So there's a question about the theoretical actual conclusion that reading non-words is the same as reading novel words. And I think, yeah, it's missing a training component to that study. And that's a critique I also share.
It can be pronounced.
Mhm. Yeah. Yeah.
I guess that's
Yeah, whether the words can be pronounced also. Yeah, very valid points. I still do have some qualms with the conclusions they draw on that paper, but I do think methodologically it was the first time I had seen something that cleverly implemented, and it opened my eyes to a lot of possibilities. So that was more so the point is their methodology. Then we'll ignore the empirical justification for just a moment.
So did you accept it or?
Eventually, yeah. Okay. Let's move into the next chunk. We're continuing on this theme of now best practices for pupil data preparation and analysis. This is going to be building on some of those same things. We were talking about nuisances, things that make it hard to design your study when you want to look at certain research questions. We also need to think about this when we get towards the stage where we're going to be preparing our data. And the reason this can be very challenging is because pupillometry is still relatively new. Best practices are still being determined and developed. I have one paper I'll be presenting this section, which is a sort of a methodological paper. And anytime I talk about this, like right now, I'm immediately like, I should be doing more because there is so much room for really demonstrating that the things that all of us who are doing pupillometry are commonly defaulting to, we need to demonstrate that those are like the best way to do it, as opposed to this is how everyone before me did it. And so some of that is still missing. The paper I have here by Wynn and colleagues, it is a very helpful starting place. It is a crazy long paper, but you can skip around and they go through like all of these different considerations, things I had never even thought about before. So it is a very deep dive for someone starting to develop their pupillometry research. My advice does deviate on some key topics, which I'm going to clarify here. So if we go back to our 3 primary nuisances, we're going to start by adding another one to this list. And this is time across an experiment. What do I mean by this? Well, when we look at pupil data, what we will see is that in the first part of an experiment, pupil responses will be larger than towards the end of an experiment. This is often referred to as the fatigue of the pupil response, but I want to clarify that it isn't entirely a fatigue response. It has been proposed that this is due to habituation to the task, boredom in the task as well. And the sort of big picture thing here is that this is a real big bummer because in our experiments, we are typically doing something like 100 trials. And if we can expect the pupil, the amplitude, the responsiveness to decrease over time, this, depending on your design, could impose a confound. So I want to very briefly show you an example from a paper that I was part of that was published in 2020. My research focuses a lot on how we perceive accents and how we get better at understanding accents the longer we listen to them. And a lot of this work has been done with behavioral data, where we have people listen to sentences with a foreign accent and write down what they heard or type what they heard. And then we see how they get more words correct the longer they listen to the accents. People get better over time. And that's been very much demonstrated in also other behavioral paradigms as well. We wanted to look at this with pupillometry. And so we developed a task where participants had 50 trials where they listened to a first language accent. So these were American listeners. So the first language accent here is American accent. And we compared it to 50 trials where they were listening to Mandarin Chinese accented English. So this is a foreign accent. We counterbalanced. There were 2 blocks where you heard 50 trials in a row from one talker and then the other. And so it was counterbalanced across blocks. And what we saw in our data was that over the course of 50 trials within the block, the pupil decreases in amplitude over time. Now, what is really tricky and was difficult about this study is that what we wanted to demonstrate with pupillometry is that the cognitive load required to process this accent is decreasing over time. As you start with sentence 1 and you go to sentence 50, because there's 1 sentence per trial, by sentence 50, it should be less cognitively demanding to process this accent because you have adapted to it. And this has been what's essentially been concluded from behavioral data. But how can we draw that conclusion when we know the amplitude of the pupil response is going to decrease over time? And the solution we came up with, which is okay, but I still think lacking in a couple of ways that I'll be quite honest about, is we had this first language accent to compare against. So here, the difference in the rate of this decrease is significant between these 2 conditions. So it's an interaction here. So we had trial, what is this sort of bins on the right side here in the legend. We had trial and we had an interaction with the effective accent. And what we found is that the amplitude was decreasing faster for the L2 accent condition. And so from that, we were concluded that cognitive demands were decreasing. But in general, we expected the pupil to decrease over time. What's sort of tricky is the L1 accent condition still isn't even the best control. So when I'm being really honest and reflective of this work, when we listen to any speaker, we adapt to them and they get better to understand. So this isn't to say that on the left, we have 100% just a pupil, like a physiological fatigue effect or habituation effect. It isn't to say that we can essentially say, because there's an interaction, we are only observing adaptation to accent here. It's still a little complicated. So this is an area where I do a lot of research and I wanted to use pupillometry. And so after this study, we were like, we really need to figure out a way to get rid of this fatigue effect. And so we're going to go back to that Win et al. Paper. This is the best practices paper that I recommended. And this came out in 2018. And on the topic of this issue in pupillometry, what they said is that you can offer breaks from the experiment to your participants. And they said for experiments of up to 1 to 5 hours, first of all, that fatigue effects weren't a big deal. This is not accurate. I hate to contradict Wynn and colleagues because I do like their research in many areas, but this is just not correct. They suggested that if you broke up the experiment with breaks, and they specifically said, chat with the participant or have them do a brief, irrelevant task, this will reduce fatigue effects. Well, they said it would improve the data in the same context as fatigue effects. That's a great idea, but we wanted to know if it would actually work because that would be great for our research question. If we could get rid of that fatigue effect, we could look at adaptation to accent so much better. So we developed a methods paper to look at this. And the title gives away a little bit what we found. Give me a break, unavoidable fatigue effects and cognitive pupillometry. But what were our aims in this study? We wanted to see if we can get rid of this fatigue effect so that people who want to look at change over time across trials could do so. And also just a note on fatigue effects, because like I said, we call them fatigue effects in the literature, but they're habituation, boredom.
I found this on the web.
Essentially, the way this sort of gets summarized is that as participants become disengaged with a task due to a combination of fatigue, habituation, and boredom, tonic activity will steadily drop, and the phasic responsiveness, this task-evoked response is our phasic response. I actually haven't used that keyword yet, I'm just now realizing. But the task-evoked response is a phasic response. And so the proposal as to why these effects happen is because our tonic arousal is lower, and so our responsiveness to in-the-moment stimulus is also reduced. And that's just going to be relevant to interpreting some of the data. So our questions: do breaks actually help? Do they actually reduce these quote unquote fatigue effects? And also, does the type of break matter? Because in their paper, they said they were going to chat with the participant. We're like, well, that's a lot of work. We don't really want to chat with the participant. We would much rather just like stick them in a room and have them do some other task. And so, I mean, I'm sorry, but I'm just not that social. So what we devised, we had a task where the participants are going to do 80 trials total. This is a speech perception task, so it's just a difficult speech and noise perception task. And so every 10 trials, we put in a break, or no break, actually. The control group gets no breaks. They go straight through. We have what's called our social breaks group, where our poor research assistant had to take these participants into a room, and she had a list of questions like, what are your hobbies? And chatted with them for a minute. The other thing we came up with, which was, I think, very fun, I don't remember whose idea it was, so I'm not saying it was my idea, but it was very fun, is what we called our kinetic breaks, where we gave participants a selection of toys. And so, for example, the toy that got used the most is kinetic sand. So I don't know if you know what kinetic sand is, but it's like this really satisfying sort of sand Play-Doh mixture. People loved it. And that's going to be relevant to the data. I'm not just saying this for fun, but like people were excited because they would come into the experiment if they're in the kinetic breaks group. And I mean, well, in any group, they were told what the process was. Either that you're going to get 80 trials if you're in control, or every 10 trials, we're going to chat with you, or every 10 trials, you're going to play with these toys, and they would see the toys before the task. So this is what we came up with. You know, in hindsight, would I have done things a little bit differently? Maybe, but let's look at what we found in this study. So this is showing you across quartiles. So I want to clarify in the analyses for this paper, trial number is our change over, or is our time, you know, our change over time measure. And it's a continuous variable, 1 through 80. But for visualization purposes, in all of our plots, we broke it into lines showing you the quartile of the experiment. So the first quarter of trials, the second quarter of trials, and so on and so forth. What did we find? So when you actually look at the models from this data, social breaks significantly reduced the quote unquote fatigue effect. However, what's really important here is you can see the fatigue effect is enormous, no matter what intervention we did. So meaningfully, we did not remove this fatigue effect. For our research question, going back to the accent question, I can't suddenly give people social breaks and not have to worry about the decrease of pupil over trials. It is still a confound in my data. It makes it really hard to do that kind of research approach. Also, just as a side note, sort of related to what we were talking about earlier about how motivation affects the size of the pupil, people who were shown they were going to get to play with toys started out with a larger pupil response. And so somewhat ironically, the people who we gave those toys to play with, although they reported that they had a much better time, they actually had a larger fatigue effect. When you look at the difference from first to fourth quartile, it is larger than in our control group. So this is important to note because, you know, when it all had said in their paper, chat with them, give them a brief irrelevant task, but actually these things can impact your data. And so at the very least, you need to think about this because if you're going to offer breaks to your participants, they need to be consistent, especially across experiment groups. Yeah. It took 40 minutes, I believe. It was 80 trials. It's a sentence perception task. They're hearing sentences and noise and then repeating what they heard aloud. Yes. Oh, so the question is, what if we ask participants to close their eyes during the break? I'm not sure how effective of a break it would be, or if it would be more effective than these. I don't have any reason to believe it would be more effective. I will say that in Win et al.'s paper, when they brought up the whole chat with the participant, it has to do with this thing called HIPPOS. And I'm going to refer you to Win et al.'s paper because they, if you're curious about this, because they have a nice plot where essentially sometimes participants, for reasons that we aren't really sure about, will start to get into this sort of rhythm of pupil response that's unrelated to anything that's happening. It's not something with eye-link technology that you can really keep an eye on during the task. But with some technologies, maybe Toby, because when Win et al. Write about it, you can keep track of it. And if you notice that the pupils essentially what's the word? Yeah, oscillating. Then you can essentially, if you alert the participant, and that they were proposing that interacting with the participant can break them out of this sort of state of HIPPOS. I've never been able to monitor for HIPPOS before. I thought it's an interesting proposal, but I don't know that that's a standard thing in pupillometry, just to clarify. It's just something that related to this paper was brought up. So if you are noticing that, closing the eyes might help, but I'm really not certain.
So, what you said from the paper that the previous paper we were looking at, they said that 1 to 1.5 hours you shouldn't worry. In your study, it seems like you should start worrying even after 10 minutes, right? You see the difference. That's statistically.
Yeah. Yeah.
So anything that's longer than 10 minutes, you would have some fatigue effects.
Yep.
And I just also wondered how much is the difference between age groups?
Oh, so that's a great question.
So when should I start being worried if I want to do pupillometry?
Okay, so this is an excellent point from Oliveira, just to repeat for people online, that this affects even within 10 trials, we're seeing it, which is accurate. And she's also asking about different age groups. I do not know any comparisons across age groups. Even this was all young adults, I should clarify. And I've done research with young and older adults. I'm going to talk about that in just a moment. But I have never seen anyone compare this quote unquote fatigue effect. I think that's a really great question though. There's a lot that still isn't fully understood. And I find this paper, like, I'm glad we did it because we sort of showed like, hey, you can't just assume you're going to get rid of this effect. And that was important, but also like, there's a million different versions of breaks you could do. And I do find it a little dissatisfying, essentially, when I look back on it. And what we concluded was that their suggestion technically improves the data, but not meaningfully. And essentially, what I'm going to recommend, and this will come up when we do practical examples of data tomorrow, is that for all pupillometry studies, time across the task, so that's your trial number, should be put into your model, essentially as a covariate. You can just put it in as a fixed effect if you're doing mixed effects modeling, but it should be put in every pupillometry model because that will control for that. If you have multiple age groups, you have young, maybe like even infants or, like different age groups, we'll just say that. And you put trial in, you can also put the interaction of trial with your group as well, and determine whether there is an effect of that. If you do that, and what you're not interested in trial number for your research, you're interested in this manipulation you've done. If you have those in your model though, it will be accounting for that variance, and it should allow you to look at the thing you are interested in better. Does that make sense? Cindy?
Like very quick, usually typically in behavioral studies, you put in a practice trial before we go into. Are these sort of built in, or does it also mean?
So practice trials is the question.
The task?
I always have practice trials and I always throw them out. And I think that is standard practice. That's what Wynn and colleague recommends, because usually the first handful of trials, the participant is getting oriented to the task quite literally. there is an orienting pupil response, the data is going to be very messy from your first few trials. This is another thing that, like in my research, like if we're looking at getting better at accent, it's a little bit of a bummer if you're throwing out your first trials, but it's standard to throw out your practice trials. Manu, how to, oh, okay. Mhm.
Were the participants doing each, or you had like control group,
Oh,
kinetic breaks? And sort of
there was
like
random assignment.
random
Yeah, and I
assignments?
believe it was 30 participants per each of
Okay,
these
so they
manipulations.
didn't choose, like, you
No.
didn't ask them
Nope.
which one they wanted.
Yeah, and the people who got the control, they never, like, anyone who didn't get to play with the kinetic sand, they didn't even see the kinetic sand.
Okay.
They didn't know they were missing out.
So they were
Um,
like anticipating.
no.
Actually,
No, but I do love the kinetic breaks group. It just cracks me up when I look back at this data and it's like, they were so excited. Like, so, Manu, did you have a question?
I have a technical question, because you said that what we are looking at here is the difference compared to the baseline.
Mm-hmm.
What's the baseline?
Yes.
So it's trial by trial,
Mhm.
because in that way you should get rid of it.
So this is a great question. Manu has brought up the baseline. We actually do look at baseline in this paper. So essentially, when we plot this, we're looking at the change on this y-axis. But before the trial, we are getting a measure of the size of the pupil and we're subtracting it from the rest of the trial. That measure of your pupil size before that phasic pupil response to that evoked response, that should be an indication of your tonic arousal level. Our data was honestly very noisy and we did not find much. We did find a trend in the predicted direction with the baseline values. But I don't remember if it was significant, to be completely honest. But this does bring up a very good point, which is that the size of the pupil before your response can be informative and meaningful. And in fact, with more complex statistical methods, you can actually take your baseline values for every trial and put that in as an effect in your model. And I have seen that in some papers, and it usually accounts for a good amount of variance. So the size of the pupil before this phasic response is going to be to some degree related to the size of your phasic response. And that's exactly, in theory, what is happening in this paper. It's just our data was a little bit noisy, to be completely honest. Let's move forward because I don't want to get too far off schedule. We have another nuisance that I wanted to really make sure I had the opportunity to tell you all about, because I know many people in this room will be interested in not just looking at young adults, but maybe looking across ages. And there is a very, very important difference between young adults and older aging populations. So this is referred to as age-related, or it's senile meiosis is the word. This is an age-related muscle atrophy. And essentially, this is just a quick snapshot from a study that's much more like physiological in nature, just showing that older adults here in the orange tend to have smaller baseline pupil diameters. Additionally, if you look at across age here on the x-axis, the peak response, the minima and the maxima, and the latency to peak, there are relationships with age. It's long story short, as we age, our pupil response changes. So you can imagine if you want to compare young and older adults, eek, that's going to be sort of hard. There have been some proposed solutions to this that I am going to criticize. So not to say that these are incorrect, but to say that we actually at this stage do not have very sufficient methodological data to support them, which is a very important consideration. So what has been proposed, this is not something that Win et al. Came up with. This is something they cover in their review paper, and they have a nice graphic that I wanted to use here. If you have a participant and you get a measure of the absolute minimum size their pupil can be, their lower limit, versus the absolute biggest size you can possibly get their pupil to be when they're in the lab. And you have one participant here on the right whose range is smaller versus a participant on the left whose range is bigger. And then you look at a evoked response here, and the difference between the peaks as you can see, essentially what they've come up with in this graph is like, how do we interpret it if the person on the left has twice the size difference, but their range is also different? This is a question that keeps me up at night, because it raises the question of, should we be controlling for what's called dynamic range across all participants? And in particular, if instead of just calling this 2 random participants, we assume, based on data, young adults are going to trend like the left and older adults are going to trend like the right, do we need to essentially, if we're going to compare these 2 age groups, do we need to convert these values so that essentially, if you do a percentage change, is what we're going to get at, then we would say this is the same change across these 2 participants. It's just this participant had less room to move, which is a very reasonable proposal, but there are some issues with it. So this was actually developed by Picquato and colleagues. It was published in 2010. And so they call this, or I'm going to call it, the light range normalization approach. The dynamic range, the maximum pupil size you can get from a participant versus the minimum, is calculated by having participants look at a computer screen that is either dark or very bright. And then a formula is applied to all pupil data within the task. And this is the formula. What this formula means is the pupil diameter at a given time point is essentially normalized by getting the range, the maximum versus the minimum, and also subtracting it up top. And you might be looking at this and thinking, what does this formula do exactly? How did they come up with it? Because that's what I thought when I saw it. I was like, well, surely they wouldn't have just pulled this formula out of nowhere. What they say in their paper is that they plotted these data based on absolute pupil size yielded the same shaped curves, but showed the younger participants as having larger pupil sizes at each point on the x-axis relative to the older participants. So to summarize what that means, the authors inferred reasonably from a host of behavioral data that the task has to be harder for older adults. So they found a way to transform their data to match that assumption. And then they concluded from their pupillometry data that the task was harder for older adults. They might be right, but that logic is very circular, and it's not a good justification for that formula. But I think, unfortunately, anyone who is comparing young and older adults since 2010 has defaulted to using this formula. And it's just the standard, essentially. And there isn't a ton of people comparing young and older adults, but there's a good amount. So I'm going to talk to you about, I have only done 1 paper that compared young and older adults and what we did about this and what we ended up sort of concluding ourselves. But mostly the theme here is going to be that we don't have a good solution to this. And I'm going to very, very much caution you against comparing these 2 age groups, given that we do not have a methodologically sound way to do so at this point in time. In this study, our aim was to investigate cognitive load for spoken word recognition. We wanted to compare young and older adults. We're going to be looking at a neighborhood density effect for those of you who are linguistically minded. We were skeptical going into this of the Picquato formula, and so we came up with 2 ways to try and explore the difference between young and older adults, trying to essentially, what's the expression, hedge our bets if the Picquato formula seemed like it didn't work well. So we decided we would do that light range normalization task. We would shine a bright light from a computer screen at the participants, put a dark screen, and get that range value, and do that formula exactly as it was in Picquato. We also developed what we called a cognitive sort of baseline task. I don't want baseline here to be confused with baseline values. What I mean here is it's a reference task. So it's another task in the study. So we are looking at word recognition. That is our main experiment. We developed another experiment that was auditory, and it's an oddball tone task. And so I'll show you in a moment what that task is like, but essentially you're discriminating high and low pitched tones. It is very, very minimally difficult. It's super easy, especially even for older adults. We weren't really sure if we'd be able to take that task and use it with a formula like Picquato's to redo our values. So this was very exploratory. So I want to be very clear that this is very exploratory. So participants do this light range task where they spend 10 seconds looking at a screen at these different light brightness. And we actually only compare the dark and the white screen. It's just so that they don't go straight from dark to white. Then they did this task where they're listening to tones. They're hearing a bunch of low tones. And then every now and then there's what's called the oddball, and it's a high pitch. And when they hear the high pitched tone, they need to click on a foot pedal. So we throw out all of the trials with that high pitched tone where they click on the foot pedal. We keep the trials where they heard a low pitched tone and they inhibited essentially their response to click on the foot pedal. It's a very, very easy task is the thing I really want to drive home here. The word recognition task, they're hearing words, they have to repeat it aloud. The words come from dense and sparse neighborhoods. I'm not going to go into too much detail here. Just the linguistic sort of theoretical part isn't the most important part of this study. For the purposes right now, for illustrating this point. So first of all, light range values, exactly as we would expect based on our knowledge of physiological data. Young adults have larger ranges, and older adults have smaller ranges in how much their pupil is able to change. This on the right is the data without the special per quadra formula. So I'm calling it unnormalized data. And what I want to show you here is that the pink lines here, this is from our word recognition task, comparing our older adults with the solid line and the younger adults with this dashed line. And you can see that older adults have larger pupil response than younger adults here, and that is significant in this study. In our oddball tone task, which was, like I said, exceptionally easy on purpose, the younger adults have larger pupil response than the older adults. What we did with this data, which, I'm going to discuss the pros and cons here. I'm not saying that everyone should do this necessarily. We determined that, first of all, older adults were having larger pupil response than younger adults without using Picquato's formula. And also what we essentially did is we didn't do any sort of special normalization, but we did point the reader to this tone task to say, as you can see, younger adults in a very simple task actually have larger pupil responses. So if anything, in our word recognition task, we are underestimating the size of this age effect. Does that make sense? And the reason we did this, and this is the final data set we reported. We wanted to err on the side of not having a type 1 error. That was like our number one goal. We didn't want to produce a type 1 error by using this formula that we don't feel is very verified. I want to show you what the data looks like with no normalization versus light range normalization. Start with this bottom graph here. This is our age effect. So this is what I just showed you, that age effect with no normalization applied. And on the right, we have Picquato's formula applied. So this is what I mean by the final data that we reported, because this is in the supplemental. We were very transparent that we did it both ways, but we said we're going to report as the final data set the more conservative estimate. This effect is freaking enormous once you apply Picquato's formula. Also notable are within subject manipulation, that density effect, which I know I didn't explain in a lot of detail, but it's just dense words should be harder than sparse words. That effect gets bigger as well. So this raises some questions. And this is just this is a strong cautionary tale. Picquato's method successfully increases the size of this age effect, wildly increases. It also increases that within subject effect, and it's a little unclear whether it should. In sum, this light range normalization increases effect sizes. However, there are some unsolved issues here. It is possible that the size of this age effect is being artificially inflated, and this means we are risking a chance of type 1 error here. And the within subjects effect may have also been artificially inflated. It is unclear whether we essentially, with this formula that's supposed to correct for age differences, whether across all subjects collapsed, whether we should have increased our within subject manipulation. And so both of these, the big cautionary part is just type 1 error. We want to avoid type 1 error. And so the conclusion we drew, we ended up reporting our unnormalized data, which still had the effects that we predicted. And we reported in a supplemental the other way of doing it. And we talked about it in our discussion that we aren't sure which is right. We aren't sure if Picquato's formula is justified. And yes, I see questions. Let's go to questions. Sandy?
I'm a bit confused about the like normalized idea because my pupil can decrease or increase differently based on light changes
Mhm.
versus when I'm listening to
Mhm.
a task, right? Because we're saying that you showed us that there's the low level going up to the high level.
Yeah.
And
So,
they're not the same thing, right? So if I'm looking, I can have, for example, my eyes can decrease or increase based on light information, but then they could do different things when I'm listening to linguistic information, right?
yes, so Sandy's question, what it is getting at, which is another thing that we discussed in our paper and I don't have a good answer to, because there still needs to be a thorough methodological investigation. I don't know that even the premise that we can use the range of pupil size elicited by light
Yeah.
is exactly maps on to the range of pupil that can be elicited by a cognitive response. We do not know that to be, yeah.
Problematic.
Yes,
And I was wondering if I do like the idea that we do need to sort of normalize it by participant.
Mhm.
There's no way of normalizing it per participant where we have like something going from high or low cognitive. It could be a digit span, what you
Yeah.
presented earlier.
Yep.
Where we could get
so that's
different
actually,
ranges, and then by that do the linguistic, something like that where
yes, Sandy is proposing like, what if we had like a digit span? So we could get essentially a really easy to a really, really hard task for this participant. I think that's a great idea. And I think as a methodological study, we could try that out and see what that, if we normalize based on the cognitive elicited range. I would love to do that, actually. That's like been on my mental to-do list for quite a while. That's sort of what we were trying to get at with the oddball tone task, but as I said, we only had the easy version here. And so I think what you're suggesting of having like a full range could be very helpful for getting at what is the proper way to do this, where we can ensure we aren't overly inflating. Yeah.
The first, at the beginning, the formulas to be able to remember, it appears to me to be like a mean maximalization.
Yeah.
Thus, the y-axis over your plots are above the beginning. So I'm a little bit worried about, not worried. I don't understand then which max and which minimum are taken into account for that analysis.
Sorry, the min and max here comes from the light range task. So if you have them stare at a dark screen, you get the mean pupil size when they look at a dark screen. And then at the light screen, you get the mean pupil size, and those values get put into this formula. The DM, I think I have this on the proof, is every single time point. So remember, your data is being collected over time. So every single time point is getting normalized by this light range value.
Yeah, the problem is the normalization is not given over the correct data, balance data, because if you normalize the trial, then you get 0, 1, and you get the delta given the population, right?
To summarize for the online group, Marco is noting some, I think, very accurate criticisms of the math, the logic behind the math here. And as I said, what Pequato was saying in their paper is they came up with a formula that made the older adults have a larger pupil response than the younger adults. And so it's possible we could come up with a better formula that's more mathematically grounded. And that could also be a solution. I think there's my main point here, and I know this might be frustrating because you're all new to pupillometry, is just this isn't figured out yet. And this is a very important consideration. Cesar?
Yeah, I mean, it reminds me of what I was saying more or less yesterday in the lab meeting, that with the cerebrovascular reactivity response, what we are trying to measure is the maximum signal in the fMRI that a subject could have.
Yeah.
So there is no measure to establish, okay, this is, I mean, I guess the minimum could be more or less estimated somehow, but is there anything that you say, okay, this is the maximum pupil diameter that you could have for this particular subject? I mean, what is the task that?
There isn't one. There isn't a standard task for determining the maximum cognitive response a individual participant can have. Not that I am aware of. There isn't any sort of standardized check for that.
Well, I mean, you know much more, but I guess that should be something that ophthalmologists should have addressed somehow, like whether the pupil can dilate or not dilate, and what is the maximum, so
Yeah.
it seems like surprising.
Yeah.
And then my other point is like, has anybody tried to combine the fatigue effect with this effect? In a sense, it's like,
Mhm.
okay, you need to normalize, but I guess the maximum and the minimum
Mhm.
also change
Mhm.
during the course of your experiment somehow, no?
Yeah.
So, and my question, like, do you intermix calibrations? I guess that's not a common practice. Like,
No, the calibrations aren't typically intermixed because they're usually the 9-point calibrations, which is more focused on eye tracking. If you are doing concurrent eye tracking.
No, I mean calibration in the sense
Oh,
of,
yes, yeah.
of estimating these values
Yeah,
during the course of an experiment.
yeah. I haven't seen that done, but that would be a nice, I think, what they said is combining it with the fatigue effect research could be important. And I think that's correct. The baseline pupil size changes over time, and this is related to the phasic response. And all of this is related to essentially this estimation of our upper limit and how that might be changing over time as well. Yeah, so there's a lot of room for methodological research for sure. And right now, if, you know, as is the standard, if you aren't controlling for differences in dynamic ranges, what you should expect is there's going to be a lot of variation. And this is why I'm going to encourage larger sample sizes, but larger is a relative term. I have within subject manipulations, my sort of default lowest has been 50 participants. And there are a lot of papers, and I do peer review many pupil papers where the standard is 30. And I think because there is so much individual variation, it's important to get larger samples, even though it's physiological data. And I think a lot of the times people use that to justify the 30 range. I do think that there is a lot of benefits given we know how much variation there is in the pupil response. So I'm gonna wrap this up. Can't tell, are we like half an hour behind? Oh, so sorry. Okay, so I'm just going to very briefly say, I put another list with a bunch of citations here at the end of this PowerPoint, which you can come back and look at. We'll talk a little bit tomorrow about how we baseline the pupil when we're processing data. I have been using subtractive baselining for as long as I've done pupillometry. There is some argument in favor of proportional baselining. Should it be a % change that we're plotting on that y-axis? The paper that led me to use subtractive baselining is cited here. It's Riley et al. 2019. But there are some people who really argue for the proportional, and so we'll discuss some of the pros and cons of each and how it affects the data. There's one of the things that drives me crazy about iLink is they give you arbitrary units. There is a discussion in this, this is a newer best practices paper that I didn't have time to read before this workshop. I only just discovered it, but they do in that paper talk about converting arbitrary units to millimeters. So you can look at that if you are interested. The one thing I will say about that, and that they say in that paper as well, is that it isn't necessary when we're thinking about within a single experiment, comparing conditions, these arbitrary units that the eye tracker gives us are perfectly fine to be using. The reason you might want to convert to millimeters is because when we think about overarching sort of like moving forward a field and being able to compare across papers, it would be nice to have millimeters. That being said, I have never converted to millimeters because I'm skeptical that the way people have told me they have converted the iLink millimeters is correct. So I sort of just wish iLink would give it to us in millimeters from the get-go. And apparently Toby does that. Other rapid fire things. We're going to talk about blink interpolation. This is filling in sections of missing data because when you blink, your eyelid covers your pupil. So we can't collect data from the pupil. So we're going to talk a lot about blink correction tomorrow. There are different methods, and you can see discussions about in favor of different methods in these papers. Downsampling data is standard. in pupillometry. And if you don't want to read this entire paper, they argue that 20 hertz is sufficient. I've been doing 50 hertz for a long time for sort of arbitrary reasons. The eye trackers typically record at 1,000 hertz. And actually, there is some very good data. So this is the Van Riege et al. Paper 2019, that there is such a thing as too much data, actually, with pupillometry. You start to risk type 1 error due to autocorrelation in the data. We'll talk about autocorrelation tomorrow. So downsampling might sound like a bad thing. It's actually a good thing for avoiding type 1 error. And then the last thing, there is a paper from my prior mentors that I felt the need to plug here, where they looked into what's called time window selection. So when we analyze with curve fitting models like growth curve analysis, which we'll talk about tomorrow, and we select a window of pupil data. So for example, this pupil data we were looking at, if I choose to analyze data from here to here versus just this little chunk right here, how will that affect my outcomes? This is essentially, it's a nice paper where they essentially took a data set that was published and they just had a computer automatically generate all of these different time windows and went to see how often the same results were found. And the answer was the results varied a lot, which isn't ideal. And the argument is essentially there are researcher degrees of freedom where people could cherry pick windows to analyze. And so this is an argument in favor of pre-registration of the window that you plan to analyze so that you can't fall into this trap. Manu?
A lot of things is very similar to EEG.
Okay.
So, and EEG is not a young technique.
Yes.
So
That's great advice.
everything that is- can be applied to it. It is in the EEG field as
Yeah.
well. So using maybe the same method for the analysis can be.
Yeah.
advantageous.
Yeah, Manu is pointing out that EEG is very similar and they've already made some of these improvements in their field. So, yes.
I'm wondering if there is pupillometry research being done with blind individuals who still have light detection.
I do not know. Look into that. Okay, we're way behind time, so I want to